Good morning, one and all present here. Myself, Dr. Pranita Gulhane, on behalf of the organizing committees, Shri Shivaji Education Society, Amravati's Science College, Congress Nagar, Nagpur, in association with Microbiologist Society India, on the occasion of 124th birth anniversary of Founder President Dr. Panjabra alias Bhausaheb Deshmukh, we are organizing a lecture series which is based on the theme Indians speaking from abroad. Today's lecture is lecture 18, which is on the topic entitled The Next Pandemic Antimicrobial Resistance. Today's eminent guest speaker is Dr. Sandeep Patil, research scientist from Department of Hematology and Oncology, Pediatric Research Institute, Shenzhen Children's Hospital, Shenzhen, China. So I would like to welcome our eminent guest speaker of today's guest lecture, Dr. Sandeep Patil sir. On behalf of the organizing committee's honorable principals, Professor Mahendra Dhore sir, Science College, Congress Nagar, Nagpur, as well as Honorable President, Microbiologist Society India, Dr. A.M. Deshmukh sir. And also I would like to take this opportunity to welcome all the participants of today's guest lecture. Now, with immense pleasure, I would like to introduce our eminent guest speaker of today's guest lecture, Dr. Sandeep Patil sir. Dr. Sandeep Patil sir is currently working as a research scientist 1 in Professor Pickwen's research team since 2018 and FLS London since May 2019. He has more than 12 years of experience in medical microbiology on antimicrobial resistance and public health, as well as the host pathogen interactions between them. More specifically, he is interested in understanding the evolution of antimicrobial resistance mechanisms in bacterial pathogens and molecular epidemiology, based on community or hospital acquired and host pathogen interactions, which help the development of new antimicrobial molecules or antibiotics. Since he started his PhD, he has been leading antimicrobial researcher, followed by a first postdoc from Zhongshan School of Medicine, and second from the Chinese Academy of Sciences, Shenzhen Institute of Advanced Technology, Shenzhen. He started his career in medical microbiology and successfully integrated this background with extrinsic academic research. He has published more than 37 publications in international journals and many plenary talks at international conferences such as Association of Microbiologists of India 2016 at Boston, USA, 12th Asian Pediatric Association, and the 17th Congress International Infectious Diseases in Malaysia. His research articles are mainly published in highly reputed, pre-reviewed SCI journals such as the Lancet Microbes, having impact factor 86.208. The Lancet Infectious Diseases impact factor 71. Clinical Infectious Diseases impact factor 20. And Emerging Infectious Diseases impact factor 16, as well as International Journal of Infectious Diseases impact factor 12. The major contribution is the discovery of colistin resistance plasmid-borne MCR1 and its transmission from animal to human, and has the potential to be used in the policy making by the Chinese government that bans colistin used in agriculture. Colistin is the last drug of choice to treat the carbapenem resistant pathogens. In addition, AMR surveillance in pediatric patents, ESBL producing E. coli, a domestic reservoir Shiga toxin, is popular work to his credit. Currently, he focusing on the ribosome targeting antibiotics, resistance mechanism, and further in-depth understanding of the mechanism of transfer plasmid between bacterial pathogens, specifically from WHO priority list. He has been awarded as Melinda and Bill Gates Young Scientist by the International Society for Infectious Diseases in 2020. Also, he has the member of several scientific and professional bodies for such as the Microbiologist Society India, as well as European Federation of Bacteria Biotechnology and European Society of Clinical Microbiology and Infectious Diseases. Thank you very much, sir, for accepting our invitation. So on behalf of the organizing committee, we are very much grateful for your precious guest lecture that we could be able to arrange today. And I would like to request you on behalf of the organizing committee that please proceed for your guest lecture. And before that, please elaborate your journey from India to abroad. Please, sir.
Thank you so much. And this is a very brief introduction. Thank you so much. Myself, Sandeep Patil, I'm working as a research scientist in the Chinjan Children's Hospitals. Mostly I work with the Professor Wen team. He is one of the member of the Health Commission of China. So I have finished PhD in the, basically I work in the PhD, I work in the Staph aureus resistant mechanisms. When I shifted to the Johnson School of Medicine, I shifted my interest from the positive to negative bacteria because it's highly resistant and easily acquired infection, so many diseases. And then continue my research in the second postdoc program. It's funded by the Chinese Academy of Science with the Professor Pagan. It's one of the renowned scientists in China. And then I started my, from last 4 years, I just working as the research scientist with my small team, connect with the Professor Wen as a main leader of my team. Today, I'm just focusing on why the antimicrobial resistance is the pandemics. Basically, the most of scientists globally, they are mentioning the silence pandemic, but from 2022, the researchers have recommended to remove the words as a silence. So the next pandemic is antimicrobial resistance. So basically my research, my topic is the mostly focus on the why it's antimicrobial is pandemic, and what it's just a background about what we are doing. It's not exactly what we are doing. It's just the overview of what we have done and what we are doing now. So why we are focusing on the infectious disease? Because if you see the scenario between the article published by the WHO and CDC, they have mentioned that the highly death occurs because of the infectious disease. If you focus on this red color, they all are they mention this death occurs globally is because of the infectious disease, they including the all bacteria, fungus, viruses. So it's a major concern of the global. So the causes of in the children's, because I'm working in the children's hospital, so I just focus on the children. So I just review the data, and we find that the overall incidence of the infectious disease has been increases from 2004 to 2017, and the target is the age of over the 10 years. They are mostly target of the infectious disease, but now we have recently one article has been submitted in the Journal of Infections after the COVID infection, after the COVID pandemic and before the COVID pandemic. So we find that the ages 4 to 10 is a highly target group of the infectious disease in the children's. So if you see the trends of the infectious disease from in 2019, the data has been published in 2001, and they are mentioned the growth of the infection. They predict 83 million people will die because of the infection in 2015. And what the effect of this infectious disease and why the infection is, what is the scenario in the globally? The recent data is published in the Lancet. They have mentioned that mostly the middle income countries. So we have just submitted one of another paper in the Lancet Infectious Disease. They have support to this one. The most of the data, or most of the countries from low or middle income countries, they have a high target of the drug resistant genes. So what is the scenario? What will happen? In 2001, they have mentioned the 83 million deaths occurred due to the antimicrobial resistant infections. But one of the nails from the USA, she has published one review and she mentioned or predict the date in 2017. They said 10 million deaths will be occurs due to the drug-resistant bacteria by 2050, and not only the impact of the health, but it's also impact on the economy of every country. It's about this about 100 million US dollar. So it's a huge economic burden, especially if you focus on the Asian countries. There's very high concern in the Asian countries. So they have another data. They just published 10 million dates. How the economic growth, how the economy will be affected by the drug-resistant bacteria. So if you see this one, they are almost 4 to 6%. If you see the 4 to 6% of the GDP is caused by the AMR infections, and this is a very great concern about our research. So why the changes from 8.3 millions to 10 millions? It's just because of the less development of antibiotics and development of resistant bacteria globally. So my target is there are several antimicrobial agents, including antibiotics, antiviral drugs, antifungal drug, parasites. But our mainly target is the antibiotics. So if you see the golden era of the antibiotics in 1940 to 1960, almost 115 new antibiotics have been discovered, and another side, the evolution of drug resistance is also started from the 1963, I think, is as per my knowledge, and they continuously increase the resistance up to 2022. But the development of the antibiotics is very less, or now is none of the antibiotics in the pipeline to develop in the coming few years. So if you see, mostly the glycopeptides is very easily develop the resistance against the glycopeptides and peptides. So why they are developed the resistance? That is the main questions. So they have the acquisition of the drug resistant genes or transmission between the people to people, or the indiscriminate use of the antibiotics. We know that everyone that what is the why they are resistance are developed throughout in the environmental contamination, healthcare transmissions, and the mass use of the antibiotics, especially after the COVID-19, they have high use of the antibiotics. So what is the mechanism behind it? What mechanisms against resistance? So mostly everybody know there's a 4 mechanisms. Modify the cell wall proteins, modify the targets production of the enzymes by some bacteria, and the efflux pumps. So mostly my target is the development of some enzymes from the bacteria, and they deactivate the antibiotics. This is my major targets. So in that my major targets, there are 2 mechanisms. There's one is the random mutations in the genome sequence, genome, or the acquisition of the genes from the resistant bacteria by the conjugation, transformation, or transductions. But in my lab, we just perform the 2 mechanisms, conjugation and transformations, mostly by the C600, the host, or by PBCSK plasmids. These are 2 mechanisms we use to transmission to study how the transmission rate of the drug-resistant genes between the cells. So how the resistance in the presence of antibiotic pressures. So when they have high pressure of the antibiotics, so how they develop the drug resistance when they have high pressures, and they transmit to the daughter cells, and the development of the resistance cells will be increased. So this is the current scenario of the global index of the antimicrobial resistance overall the world. So I'm working in China, so I just concern in this area. They are high. If you see the index is more than, it's almost a 72 to in between the 64. It means the high resistance we have in China. So we need to focus on the policies, mechanisms, and development of new molecules. So how the transmission they occurs in between the all the one, if you see the One Health program, they have the transmissions. So mostly they are transmission between the animal to the human. So this is a main concern I focus when I do my first postdoc and second postdoc, and I finding the MCR1, it's and this transmission from the human to human from the animal source. So the clistin high use in the agriculture farms, or so they develop the resistance in the agriculture and transmit to the humans. And now it's we call it as a non-treatable infections due to presence of the MCR1. So another is the transmission of the in the health and health associated environment. So we have just published one of the articles in the EDI, the transmission of the MCR1 in the transporters like tube, we call it metro. So in the transmission of the metro, when we have published this one, the government has taken a decision to stop the use the coins, because we have the coins to enter in the subway, but now they stop the coins because they are transmission of some infectious disease due to the coins. So another is when they develop the resistance, how they categorize the drug resistant bacteria, that's MDR, XDR, and PDR. So the MDR is maybe developed in the 80s. After that, they addition some drugs, resistant genes, and now we have the PDR. So if we have the any sensitive and not sensitive agents to the antibiotics, any antibiotics category, so we call them the PDR, and it's very difficult to treat now. So it's based on that one, what the WHO suggested. Suppose WHO already mentioned the priority list of the pathogens. So we have focused on these 3 bacteria in our group, to E. coli, Klebsiella, carbapenem resistant Klebsiella, Acinetobacter, and Pseudomonas. So these are the major 3. So this is our priority list, and so we are just focusing on this one recently, mostly the ESBL and carbapenem producing the Acinetobacter and Pseudomonas. So what the global action plans to follow to the drug resistance? This one is the communication, give the educations. The second program is the AMR surveillance, laboratories, and do the research. This is our main concern. We are in the research, infection, and the healthcare facilities. They are the government program to do the preventive and animal transmission stop the, and government, based on the research, the government make a policy to regulations to use of the antibiotics, and the patient and the doctors make the policies to how antibiotics can use in the veterinary and agriculture. So this is my team, and we are always surveillance programs to my team to how use the antibiotics because I'm working the oncology. So we have the mostly 3 departments. One is the transplant and leukemia and the tumor. So mostly when we transplant patient from the transplant ward to the leukemia, so leukemia, when you transplant to leukemia, this patient mostly the neutropenia, the fever neutropenia. So this very serious conditions. So we should be very careful about using the antibiotics and transmission of the disease in the leukemia patients. So another our laboratory job is to make the surveillance and to do the research on the AMR, so what is the mechanisms and how they develop the cells, and what we can do in the future to develop some new molecules based on the targets, or based on the ribosomes targets. So the first is the surveillance. So we make the data analysis from not only from our hospital, we have global networking network in the UK, Africa, and the some Asian countries, including the Japan and the Thailand and Malaysia. So we understand how the drug resistance scenario each country and make some data to report to the government. So these are some bodies or organizations. They work throughout the global surveillance. They make a surveillance. So what we do in the national surveillance system, so that's our main target, is the laboratory surveillance, the flow informations and data collections, and inform to the from national laboratories also to the government. So this is the pie diagram, pie to the local surveillance, then the national surveillance, the regional surveillance, and the global surveillance. Then we can able to tackle the drug resistance, but we cannot stop it because it's a spontaneous mutation in the genome, so we cannot stop it. But we can definitely reduce, or if we understand the target sites, so we can make a molecules to develop the or tackle the drug resistance. So these some techniques we are using in our laboratories, including the gnat or fish, to detect the MR in our laboratory. So this is another, recently we are using the whole genome sequence, or all the recent techniques we use in our laboratory. So what we found so far, that's main concern that we have find in the China. So when we are looking to data, so we find that carbapenem resistance is highly resistant, so highly spread in the Chinese community, and what the bacteria in the gram-negative. So we first focus on, like WHO has focused already. So we are mentioned as E. coli, Klebsiella, and Pseudomonas. They are the major concern. They have acquired high resistant genes, including the multiple or coexistence of resistance on same plasmids, OXA23, CTXM15, TAM, or SHV. They are highly concerned, but it's not so, we're not concerned too much about the SHV, but we are concerning about the KPC, CTX, and OXA in the Chinese community. So what we find so far, this our some work has already published. So we have already find what this is. If you see the cefotaxamine is a third generation of cephalosporin. So we have already high resistance of third generation of cephalosporin. Even we have the now clistin resistance high rate. So this is very concerned to about the in the patient when we are treated in basically mostly in the pediatric patient, because these are natural resistance they can acquire when we treat high dose of the antibiotics. And this is the distribution of some genes among these all the cells, all the bacteria. So one of the recent we have published in the Lancet Microbes. So we have suggest when you're making the PVC 20 vaccines, they may not be considered 24F, because if you see the data from the Japan and the Thailand, even though from India, we don't have the 24F strain in our Asian countries, but we suppose that we will be very soon they have be introduced in the Asian countries. So when they are making the PVC 20, because now we have the PCV 13 vaccine for the pneumonia, but when now it's in the pipeline, the PCV 20. So they should think about the when the introduction they think about the non-PCV 13 strains, that is 11A and 24B, because they have high resistance. We have reported the high resistance, and only the oxacillin is the drug of choice now to treat the S pneumonia in the pediatric patients. So another important thing, how what is the backbone of the transmission of genes? But like we mentioned, I have mentioned two mechanisms we are focusing on: the chromosome base and the plasmid base. So when we study the plasmid base mechanisms, what is the backbone of the transmission, high transmission of the plasmids from the one cell to another cells. So we are already they published the data. So they have mentioned these are three groups of the plasmids or the family. They are highly or easily transmit from the one cell to the another cell, including they carry the drug resistant genes. So if you see these red marks, they all are the MCR genes with the coexist with the CTXM. So even they have more resistant genes they carry in the single plasmids. So this is very concern to the development of drug resistance. Now we should understand the genetic environment. What is the genetic environment? Where they are the chromosomes, where they are the plasmids. How we can do the PFG, and we do the PFG, and we have designed some markers to specific genes to see whether they are where they are located in the chromosome and plasmids, and how the clonal relationship between the cells. Even after now the PFG we do it, but we do the whole genome sequence, so we no need to do the PFG again because the MLST we can find in the database. So these are the one of the newly found MCA1 is we not published yet, but it's we are finding the two E. coli strains, one isolated in the 2015 and one isolated in 2018 from diarrheal patients. And we find that these two E. coli cells have the same kind of mutations. These are the two mutations. And but the interesting is they not reduce. They reduce the hydrolytic activity, not increase against colistin. So we have make some site-directed mutagenesis and finding the how the important location of the genes, so we can reduce the hydrolytic activity in the MCR. So these are the chromosome-based MCR1s we have already reported in the general already reported, and not only we have focus on the plasmid-based or chromosome-based, at that time we are finding some novel strains. One of another we have reported one Elizabeth Kingia is non-pathogenic strains, but we have reported in the leukemia patients because she's a female, 7-year-old female, and we have transplanted, and after that she got infection, the UTI. And so we treat the different, even the colistin we have treat, but she did not recover because it's recover 10 to 15 days, but recurrent infection is occur. So these are another challenge we have facing based on the drug resistance now. So these are some we have done some whole genome sequence studies and the plasmids. So another important things in the collaboration with the Aberdeen University UK, we have one project we have just published in the Lancet Microbe. So we define because the most of children has taken the meat foods in the breakfast or the evening, mostly in the China or European countries. So we define that STEC is the ESBL encoding with the toxin genes in the E. coli 131 is a sporadic or epidemic strain reported in the USA. So this is a reservoir of the toxin genes. So this one is also just published in the Lancet Microbe. So now we focus on the molecular epidemiology. What we do in the molecular epidemiology, we mark some specific genes, and we look at the distribution globally, how they spread from the one cell to another cell by. So when we do normally we do now we just stop do the PFG because we do now we are whole genome sequence, but this is from my postdoc work. So we have collected from the different species and different environmental samples and different specimens, and to define how the conjugation type or plasmid type, ESBL type or EST type among the isolated strains. So far I have almost 7,000 strains, E. coli strains and Pseudomonas strains, so to study. So this is a major surveillance program, so in the China or in the globally. So, but my focus is on the Pseudomonas and the Klebsiella and Enterobacter species. So far, I didn't focus on any of the Streptococcus or Nigeria in my lab. So these are the major concern by WHO, and for us also, so, we are focusing on it. So the strategy 4 is the prevention and the control. So we know that everyone, after the COVID stay, they are mostly the hygienic sterilization program, vaccination, hand sanitization, biological safety. We do the same program. It's object third of the antimicrobial resistant. Mostly we just focus on the hospital transmissions because recently we have submitted one paper in the Journal of Infection, and we found that the same kind of CTXM with the similar plasmids that are transmitted in the environment, and even these nursing staff and the patients, we found a similar kind of strains, even not on the strains, the plasmid also the transmitted from the one cell to another cell in the environment. Even the health community, we can vaccinate program and cleaning the community. So the fourth strategy is to the rapid detection and the surveillance. In the rapid, we can phenotypic detection, we can do the molecular detections and to reduce the antimicrobial resistance. So another is the we can look at the synergetic effects of the mixing of two antibiotics to treat some MDR. But as we do some experiment at the preliminary level, but we didn't find so far any drug mixing of the drug or some synergetic effects. We cannot find it. So these are the drugs recommended by in 2019 by the guidelines to treat the infection. So now after I make my small group, now I focus on the non-digestive oligosaccharides to treat the respiratory infections by using the organoids. So I have some, I have collect some organoids from the infected to see the action of some oligosaccharides, which I have already we extract the oligosaccharides. So these are my next target. We are targeting this one, like ribosome targeting antibiotics mechanism study, how the tRNA and translated to translocations and the translation. When I know that the mechanisms we are next to. We have some find out some non-digestive oligosaccharides. They have the antimicrobial pathogenic activity against gram-negative organisms. So further, we are studying the in vivo and ex vivo using the organoids to understand how the oligosaccharides they break down the cell walls and interact with the chromosomes to the next so-called antibiotics we can use in the patients. So for that one, we have the collaborate in the Spain and the Netherlands to perform this work. So far, the work is after I joined the research scientist, my work as a grant, find a grant at three different organizations on the state level and even the international levels. I have getting some grants to perform the experiment, and these are my collaborators globally to perform some experiments. So I have just now one a postdoctorate and one research associate to do the run out of my lab. So thank you. Okay, one of the questions from the hello.
Sir, can you please minimize your presentation, sir, Patil sir?
Okay. Okay.
No, sir, yet it to be properly minimized.
Okay.
Yes, yes, sir.
Okay. Yeah.
No. Yet to be minimized, sir.
Now. No.
Sir, you close your presentation.
Participants,
Yeah.
please wait for a while, please, sir. Then I would like to ask some questions which are in the chat box. Yes, sir. Now it is fine.
Okay.
Patil, sir. Okay.
Yes. Okay. Okay.
Yes, sir. Dr. Prakash Alami, sir, have the question in the chat box that how serious is colistin resistance in Asia, since this antibiotic is used very commonly in clinical settings and veterinary?
No, it's not used in the commonly in the clinical settings. It's a last drug of choice, so it's not used in the clinical use, but is highly used in the agriculture. I don't know about the some other countries, but most of countries, Europe and the US, even the Japan and the I have collaboration in the Malaysia. So these governments has already banned the colistin in agriculture use. We cannot use the colistin in the agriculture now.
But now actually in India it is under use. Of course, we have the National Action Plan pertaining to minimizing the usage of the antibiotics, and colistin resistance actually we also found very extensively in the gram-negative pathogens. So,
Yeah, it's a gram-negative.
you know, after China, I think India is the next where maximum antibiotics are used, and I think even colistin is imported from China in India.
Yeah, now they're India is importing, but in China we have banned the colistin use. We cannot use the colistin in the now.
Okay. But then the actually question was, since this is from bacterial origin, not the synthetic antibiotics, why we should have this much a drug resistance problem? What is your opinion, sir?
What do you mean?
No, usually the no chemically synthetic antibiotics we see the drug resistance.
Yeah.
Since this is actually from microbial or bacterial origin. We see almost no very high level of drug resistance for the colistin also. Our actually expectation or assumption was that when any antibiotics of microbial origin may not have that much of drug resistance like the synthetic antibiotics. What is your opinion?
The synthetic drug, if you use the synthetic, it depends on the target site. Because,
yeah, exactly.
Because of the depends on target site. So that's why we are looking now, it's ribosome targeting, because most of the antibiotics from the ribosome targets. So we are looking to understand how the tRNA's translations occurs, and how we can make we have already designed some as I mentioned, we have some collaboration with the Netherlands. So we have Professor Windy, and so we have designed already some non-oligosaccharides molecules to look at the respiratory specific and the respiratory infections. So we already have some results preliminary. So we are getting some organoids to look at see the flow cytometry, how the drug infection rate is decreased in the patients. We are looking at that one.
Okay. The last question was actually we reported colistin resistance in lactics also. Yeah. Lactic acid bacteria.
Lactic acid bacteria. Yeah.
Is it transmissible or it is an intrinsic resistance?
No, it depends on where their locations.
Yeah, mostly chromosomal.
No, because the MCR1 because the colistin is resistant by the two mechanisms. Is the efflux pump is one mechanism. It is we know everyone before 2020 2016 it's a well-known mechanisms, but after our discovery we have MCR1. So I should want to know what is the mechanisms. We should maybe know the mechanism. If it's a plasmid bond, then definitely it will be transmit.
Yes, exactly.
Yeah, if it's chromosome based, it's very less. It cannot be, or it is a efflux pump, then it's not chances to transmit very fastly.
Colistin resistance you are investigating in your lab now?
Yeah, of course. We are. This is our major concern now, colistin. But now the rate is decreased, and the now government focus on the applied. How whatever we have the data now, the mechanisms so far. So how can utilize these mechanisms or these targets to investigate the new molecules to treat the MDR is currently.
Exactly. How is the carbapenem resistance problem in China?
Carbapenem resistance is almost a 34 to 39% carbapenem resistance. Because of the CTXM 14 and 15, we have high rate. KPC is not too much. KPC 55 is very less. But we have OXA 23 and OXA 22. It's major concern for us.
Exactly. You think in India also carbapenem resistance still it is spreading? Because of our poor hospital
we have NDM1, we have discovered NDM1 in New Delhi. So
because many Asian countries, NDM1 was detected.
Yeah,
almost now 10
more than
years.
India is more than China. It's almost 72% as per my data. India is a 72% of carbapenem resistance.
No, colistin problem is because it is used for the animal husbandry and in poultry also.
Yeah. Poultry is so much used in the poultry.
Yes.
So we have already defined one paper. They are transmitted from the pig to the human.
We have actually colistin resistance review in Journal of Antibiotics. I will share with you.
Journal of Antibiotics is MDPI.
Yes. Thank you.
Okay, thank you.
One more question in the chat box from Sachin Kumar. Is there any options to use of herbal-based antimicrobials as alternatives to antibiotics?
Of course, they have the base. We can use it, but they have many scientific questions about. Last lecture, I also have I asked that question. How you can purify it? How you can justify these clinical trials? How you can after the purification they have efficiency? There are so many questions. Even you find the molecules, some extract. So how you can define you just the lab is okay. You can just make one plate and so many some plant material, extract the material and just pour in the plate. That is not enough to say something. We should have the clinical trials. We should have the side effects on efficacy study, and we have the separation of the molecules, the stability of the molecules. There are so many things.
Actually, it is described in our National Action Plan in India, use of the herbal also, along with the conventional antibiotics, where we can actually minimize the antibiotics dosage. It is a good idea.
Hello.
There is one more question.
Very good morning, sir. Hello.
Very good morning.
Sir, we are working with herbal-based extracts on basically E. coli as well as Staphylococcus aureus. So we are just going with two plants. So we got good results. Antibiotic sensitivity was found. The bacteria was found to be sensitive to both plants. So we are working with and both bacteria were found to both bacteria were found to resistance to colistin drug as well, sir.
Okay.
Yes, sir.
So this colistin resistance is because of the efflux or because of the MCR?
Sir, we applied the Octadisc in which 8 antibiotics are found. So in 8 antibiotics, colistin is also present. So when we see the inhibition zone, there are no inhibition zones on both bacteria against colistin as well as ampicillin also, sir.
You mean your bacteria is the resistant to colistin? That's the first thing.
Yes.
Okay. So my next question is the colistin resistance is made up of. Do you know why the colistin resistance is because of the efflux or because of the MCR1?
So basically, just we go preliminary screening, and now we are approaching towards the mechanism behind the herbal-based products to. Yes, sir. So we just going through the preliminary screening.
That's good. That's good.
Yes,
that's good.
Yes.
That's good.
There is one question in the chat box that food and drug resistance, how it can be a fatal in future. What are its preventions and strategies?
Food drug resistance?
Just participant has written that food and drug resistance, sir, how it can be a fatal in future, its prevention and strategies.
Food drug resistant, I didn't understand. What do you mean by food resistant?
Participant can elaborate your question, please. There is not proper name given to that chat. That name is given DNA lab center something. Okay. One more question is that from Nilima Adusmali, sir, can we have combat strategy with plant metabolites? Can we have a combat strategy with plant metabolites for MDR?
Yes, we can.
Very good morning.
In the research, every possibility, we have every possibility to check it.
Participants can type their questions, rather than asking, because there is some network issues also.
Oh, yeah.
One of the participants wants to ask something, but I think
yes.
network is not clear.
From Malika madam, it is the question that, is your work complete bioinformatics based? What should be the WAG line of project for studying the AMR?
No, it's not based on the bioinformatics. We're based on the clinicals, because we are working in the hospital. So it's, of course, it's bioinformatics is one of the part of our research. It's not everything about the bioinformatics.
Okay. One question from Dr. Sandeep Kumar. What are the possibility of phytochemicals to work as an alternative to antibiotics?
I told you the phytochemicals or plant-based, everything is the same.
Same answer.
Phytochemicals is
same. What we discuss about the herbal formula.
Yeah. Herbal is the same.
Exactly.
There is one question from Madam Nilima. World scenario says AMR is maximum in Asia. How is it kept under control in other countries?
There are very strict policies about the use of the antibiotics. They are very strict to prescribe them. Like if you see the India, you can go to the pharmacy shop and you ask some medicine, they can easily give the antibiotics. But if you go to the US or UK, you cannot get the antibiotics without prescription. So there is a limitation use of the antibiotics. The first thing, there is so many awareness program run by the European Union in the European Union. So people knows use of the antibiotics and that's the disadvantage of the higher dose or lower dose of the antibiotics. Even their education about the antibiotics. So when you prescribe the antibiotics for 3 days, like a 6 dose, it should be finished in the 6 dose. It cannot be finished 6 dose. We just take 1 dose or 2 dose, and then we said, okay, we are fine. So we no need to antibiotics. It's finished.
Okay, there is one question very much interesting from the unknown participant. When is the next pandemic expected?
Now we are in the next pandemic. So already India has 72% drug-resistant bacteria. We are in the pandemic.
Started already.
It
Yes,
already started.
yes, it is really have been started.
MRA say VRE is the major problem right now, up to 78.
We should be very much careful.
Is a major concern.
Then MDR, tuberculosis.
Tuberculosis MDR is also a major concern.
Okay. And yes, one question is that which samples are collected during surveillance of AMR?
Which sample? We collect all the samples.
Okay.
We are all the specimens. When we have when we look at the samples from the water, sewage, air sampling, transport sampling, environmental sampling, even the plants surface.
All the three different ecosystems. All the three different ecosystems.
Yeah, one health program is
one health program.
One health program. If you see the one health program, they have all the sampling points.
Okay. So if the participants have any queries, then you could please ask, because in the chat box I have asked most of the queries. Okay, so thank you very much, Dr. Patil sir, for your precious guest lecture. You have really elaborated your topic, and participants also have been satisfied by your information which you have given to us. Now, I would like to request you that please put your email ID in the chat box, so that in future, if the participants would like to ask any queries, then they will definitely contact you on your email ID. And yes, thank you very much on behalf of the organizing committee's Honorable Principal Professor Mahendra Dhore sir, Science College, Congress Nagar, Nagpur, and Honorable President, Microbiologist Society India, Dr. A.M. Deshmukh sir. On behalf of the organizing committee, I, Dr. Pranita Gulhane, would like to thank you very much, Dr. Patil sir, for your guest lecture, which is based on the theme Indian Speaking from Abroad. So once again, thank you.
Thank you. Thank you. Thank you.
Thank you, sir.